Foundation Module 9 UHS Modular Integrated Undergraduate Curriculum Physiology Dr. Sheen CEO and Founder Sheikh Zayed Medical College Physiology Department. Medical Superfast Simplified Physiology Video Lecture Notes. Guyton and Hall 14 Edition Chapter Number 51 Lecture Number 10. Learning Objectives. Describe the structure of rods and cones. Structure of rods. Rods and cones are the two types of photoreceptor cells found in the retina of the eye. They are responsible for converting light into electrical signals, which are then processed by the brain to produce visual images. These cells are crucial for vision, but they differ in structure, function, and the types of light they detect. Although both are photoreceptors, rods and cones have different structures and functions. The major functional segments of either a rod or cones. 1. The outer segment. 2. The inner segment. 3. The nucleus, or cell body. 4. Photoreceptor layer, contains two main types of photoreceptor cells: rods and cones. The rods are narrow and longer than cones. In the peripheral portions of the retina, the rods are 2 to 5 micrometers in diameter. In the central part of the retina, in the fovea, there are no rods. Do not detect color and are more concentrated in the peripheral retina. Rods, outer segment. The outer segment of rods is elongated and cylindrical. It contains stacked membranous discs that are densely packed with the photopigment rhodopsin. Function: these discs increase the surface area for light absorption. Rhodopsin is highly sensitive to low light levels. Inner segment. This part of the rod is broader than the outer segment and contains numerous mitochondria, ribosomes, and other organelles necessary for cell maintenance and energy production. Function: the inner segment is responsible for the metabolic activities of the rod, including the production of ATP, which is essential for maintaining the photoreceptor's function. Nucleus, cell body located between the inner segment and the synaptic terminal, containing the nucleus of the rod cell. Function: the nucleus having the genetic material and is involved in the transcription of RNA necessary for protein synthesis. Synaptic terminal: the terminal portion of the rod makes synaptic connections with bipolar cells and horizontal cells. Function: it releases the neurotransmitter glutamate. Structure of cones. The outer segment of the cone is shorter and conical in shape. Like rods, it contains membranous discs. These discs are continuous with the plasma membrane and contain the photopigments, called opsins, that are sensitive to specific wavelengths of light, allowing color discrimination. S-cones, sensitive to short wavelengths, blue light. M-cones, sensitive to medium wavelengths, green light. L-cone, sensitive to long wavelengths, red light. Function: cones are less sensitive to light than rods but are important for color vision, photopic vision, and function best in bright light. Inner segment: the inner segment of cones is similar to rods, containing mitochondria and other organelles required for cell maintenance. Function: it performs metabolic activities important for the function and survival of the cone cells. Nucleus, cell body. The nucleus is located between the inner segment and the synaptic terminal, similar to rods. Function: it plays a role in gene expression and protein synthesis. Synaptic terminal: the terminal portion of the cone, called the pedicle, is larger and more complex than the rod's synaptic terminal, allowing for multiple synaptic connections with bipolar and horizontal cells. Function: like rods, cones release the neurotransmitter glutamate to communicate with downstream neurons, but they are involved in transmitting information about color and fine details.